One important objective in genetics is not only to identify the genes that control particular phenotypes or traits, such as drought resistance in plants, but also to map them. That means determine their location on particular chromosomes and where on a particular chromosome they are located. And this activity task is called mapping. And one important technique for mapping genes on chromosomes is called the 3-point Testcross. Before we go into the details of the cross and how to figure out the distance between genes, let's introduce some genes that we are using as an example for this 3-point Testcross. In corn, plants, wild type plants have green leaves, and it's a dominant trait P+, but there is a mutant that produces purple leaves denoted with P, and it's a recessive mutant. Wild type plants are sensitive to virus, so they're denoted as V+, but there is a recessive mutant called V, which makes the plants resistant to virus. Wild type plants have plain seed, and that's again a dominant trait denoted by B+, and there is a recessive mutant called brown midriff, and that's denoted by little p. So in a 3-point Testcross, you start with pure breeding lines, and in this case we are starting with a line that is dominant for all the wild type alleles, that is homozygous, sorry, for all the wild type alleles of the genes. And so this plant is wild type for all the 3 traits. And you cross this to an individual who is homozygous for the recessive alleles of all the 3 traits, and the recessive alleles are the mutant alleles, and therefore this individual can be called a triple mutant. And the gametes these 2 individuals will produce will be either P plus V plus B plus, and all the gametes will be the same because this individual is pure breeding and is homozygous for all 3 traits. Likewise, the gametes produced by the triple mutant individual will all be PVB, since that individual is also homozygous for all 3 traits. And their F1 progeny will be a trihybrid, since they are homozygous for all 3 traits. And in the 3-point Testcross, in the next step, you carry out a Testcross by crossing this trihybrid individual to the tester who is homozygous for the recessive alleles of all the 3 traits. And the objective of a 3-point Testcross is to look at the fraction of different progeny genotypes and use that to determine how far apart are the 3 genes on the chromosome, what is the order of the 3 genes on the chromosome. And this table shows the kind of data that you obtain in a 3-point Testcross and which will be typically posed to you in such problems. And here you are given the gametes, the genotypes of the gametes of the trihybrid individual. And so these are gametes generated by this individual, and the phenotypes of the F2 generation simply follow the genotypes of the gametes of the trihybrid individual. For example, if you are P plus V plus B plus, then you will have the dominant phenotype for all the 3 traits. So you'll have green leaves, you will be sensitive to virus, and you will have plain seeds. Whereas if you are if the gamete of the trihybrid individual is PVB+, you will have the recessive phenotypes for the first two traits. So you will have purple leaves and you will have virus resistance, but you will have the dominant phenotype for the third trait, which is plain seeds. And our goal is, given these data, to determine as a first step how far apart these genes are. And so let's get started, and we will work with pairs of genes, and perhaps we will work with PV first. And the first step in working out a 3-point test cross is to write down the parental genotypes for the pair of genes. And the parental genotypes we know from the gametes that the parents produced, and the parental genotypes are P plus V plus, that's one parent, and the other parent has parental genotype PV. The second step in solving a 3-point test cross is to write down the recombinant genotypes for this pair of genes. And that's easy once you know what the parental genotypes are. If the parental genotypes are P plus V plus and PV, then the recombinant genotypes must be P plus V or PV plus. And since the distance, the mapping distance between two genes is given by the percentage of recombinant progeny or percentage of recombinant gametes, if we simply go in this table and identify how many of the progeny or how many of the trihybrid gametes have recombinant genotypes, we will be able to compute map distance. And so the third step is to determine the frequency of the recombinant genotypes. All right, let's go ahead and identify the recombinant genotypes between P and V. And so anything which is a P plus V is a recombinant genotype, and we find that this is a recombinant genotype and this is a recombinant genotype. Also, anything which is PV plus is a recombinant genotype, and we find there are two of them there, PV plus here and PV plus here. And so the distance can be computed by adding up how many progeny or how many of the gametes are recombinant and dividing by the total number of individuals. And so that's 1024 plus 1044 plus 72 plus 60, and the total, if you do the math, is 10,000. So we divide by 10,000, and once again, if you do the arithmetic, this will turn out to be 22%. Or we can say that PV are separated by 22 map units. Next, let's repeat this calculation for V and B. And the first step, as before, is to write down the parental genotypes, which are V plus, B plus, and VB. Therefore, the recombinant genotypes are going to be V plus B and VB plus. The third step is we can go in this table and identify wherever we see a recombinant genotype for VB, which is V plus B or VB plus, and we have VB plus over here. We also have a VB plus over here. And then we have to hunt for V plus B, and they can be found here and here. And therefore, in the third step, we will compute the frequency of these recombinants between V and B, and that is 1024 plus 1044 plus 690 plus 678 divided by the total population, which is 10,000, and that gives us 34.36%, and therefore we say that V and B are separated by 34.36 map units. As a third step, we can compute the distance between P and B, and once more we start by writing parental genotypes, which are P plus B plus and PB, which means the recombinant genotypes are P plus B and PB plus. As the third step, we can go hunting in our table looking for P plus B, and we find P plus B over here and over here, and now we can look for PB plus, and that's over here and here. In order to compute the map distance between P and B, we can add all of these up. So 690 plus 678 plus 72 plus 60 divided by 10,000, and that will give us 15%. Therefore, P and B are separated by 15 map units. Now that we have the distances between all three pairs of genes, we can use that to find out in what order they lie on the chromosome. And clearly, VB are the farthest apart. And so if we had to draw a map of the chromosome, it might look something like this. There would be V and B, which are separated by 34.36 map units, and P is in the middle, and P is closer to B than to V, so something like this. And this distance between PB is 15 map units, and the distance between V and P is 22 map units. Of course, it's possible, we can only determine the relative order, and it is equally possible that B lies on the left and V lies on the right. either map is correct. One interesting inconsistency, or paradox if you will, is that if I add up the distances between BP and PV, I get 37 map units, which is more than the 34.36 map units we calculated using recombinant frequency for the distance between B and V. So which one is correct? As it turns out, it is this distance which is correct, whereas this distance is an underestimate of the true distance between B and V. In order to see why that is the case, let us draw crossovers between the two parental chromosomes. And so we have one parental chromosome which has V plus, P plus, and B plus. And we have the other parental chromosome which has V, P, and B. Now let us say that we have a double crossover between these two non-sister chromatids. And for double crossovers, I'm going to go and erase over here and here, and we will get a crossover like this. And a crossover like this. As a result of this crossover, one non-sister chromatid is going to have the genotype V plus P B plus, and that's here, V plus P B plus. And the other chromosome will have the genotype V P plus B, and that's over here, V P plus B. In our earlier calculations of the distance between V and B, we had ignored these two genotypes. Since VB matches the parental genotype, we regarded this as not being recombinant. Similarly, V plus B plus matches the parental genotype, and we had regarded this as also not being recombinant. However, visualizing these crossovers makes it apparent that in fact these are recombinant genotypes, and they are the result of double crossover events. And even though V plus and B plus haven't changed places, it's P plus and P that have swapped chromatids, and therefore these are recombinant genotypes. And as a result, we must count them when we are computing the distance between V and B. And not only should we count them, but we should count them twice, because they involve two crossover events. And therefore, now for the distance between V and B, we will add up 1024, 1044, 690, and 678, which were already recombinant, which are apparent, are recombinant between V and B. However, we must also include P plus VB, this genotype, and multiply it by 2, since this genotype was produced by a double crossover event. And also we must include PV plus B plus, again multiplied by a factor of 2, since this is also the result of a double crossover event, and divide that by 10,000. And if you work the math out, it will turn out to be 37%, or 37 map units, which is identical to the estimate obtained by adding the distance between P and B and P and V. Finally, we can use this insight to solve 3-point test crosses in a much more efficient manner. 3-point test crosses, the data in 3-point test crosses always have this pattern. So you will find two genotypes that have the lowest frequency, and you can always infer that these are due to double crossover events, since having two crossovers is very unlikely. Similarly, you always have two genotypes that are the most abundant, and you can infer that those are the parental genotypes, since even a single crossover is a relatively unlikely event. The parental genotypes are always the most abundant. And what this means is that even if we were not provided with the actual 3-point test cross, so we did not know what the parental genotypes were, we can always just look at this table and pick the most abundant genotypes as the parental genotypes. And finally, you have genotypes that have intermediate values or abundances, and those are single crossover events. Now, let's write down the parental genotypes, which are B plus, V plus, B plus, and PVB. Let's also write down the double crossover genotypes, and they are P plus VB and P V plus B plus. And it's clear that the gene that is switching alleles, there's only one gene that is switching alleles between the parental and the double crossover genotypes, and that's P, is the gene in the middle, because only the gene in the middle changes alleles, creates a recombinant genotype in a double crossover event. And therefore, we can make a map without having computed any distances that V and B must be on the outside and P must be on the inside. And then once we know the order of the genes, we just need to compute the VP distance and the PB distance, and we can add those up to get the distance between V and B, and this makes for a much more efficient way to solve the 3-point test cross.